somebody could write it, perhaps somebody could write in YouTube that this is what we're going to do so that they don't wait.
Yeah.
I'm trying to do that now.
I'm sorry for that. I should have checked it better before. That is absolutely my fault. But I mean, you see that I'm recording now. You had to click OK. So I'm going to continue here. And like I said, the last times I was watching YouTube, there were less than 10 people, and that was me and at least 2 of our students. So it was even much more less than this. And I know many people watch it afterwards, and I will then upload it then. Okay, so I go back to this. So this paper arose from discussions of the foreign audience of Peter Kapler, asking it would be nice to give an overview what data are needed, and for Filippo Aureli, especially asking for the Xenatra, these are the sloths, the anteaters and armadillos, what they should report in their papers. And then Lindelani Makuya was working with Charlotte on the Xenatra for a database on comparative mammalian social evolution. And in this during this internship, she looked at more than 4,000 publications on Xenatra, identified 62 that were field studies where people probably collected data on social organization. But out of these, only 12 papers reported the data in a way that it could be used to infer what is a social organization. And then in this paper, they come up with recommendation what people should report in the future, because we think the data collected by field researchers are at least as valuable as other conclusions. And for future studies that want to test hypotheses that need to rely on such data, it's very important that the data themselves are also reported. And to promote this, I also have to be quicker now. If you're interested, I will put the link for this paper into the chat here. And if you think it's a great idea to support this idea, then please share it on your social media, like on Twitter. Lindelani is also presenting it tomorrow at the International Congress of Zoology. That's a remote congress. And we're also very happy that this paper could be published as ecological methods in the Journal of Ecology, the editor of which is Wolfgang Goymann, who is actually our speaker here tonight. And Wolfgang is doing an incredible job as the editor of Ecology. He improved this, from my point of view, this journal significantly by introducing new topics that can be published there, apart from the ecological methods I just talked about, for example, perspectives and refuse, that we had another fine publication about using remote seminars to teach animal behavior. They have the topics on species in the spotlight and behavioral notes. So it's a very good journal, of course, also for the normal research articles, but also to publish these other ideas and perspectives in the field of ecology, animal behavior, and behavioral ecology. So Wolfgang did his PhD at the Max-Planck Institute in Seewiesen. At the time I was there a master student. He worked with Heribert Hofer on hyenas in the Serengeti National Park, measuring hormones in feces. It was about female dominance. And since then, he has been always interested in the ecophysiology of life histories. He already, when he was down in Tanzania, he got in touch with them, or he realized that there's some very interesting bird species like the Coucals he's going to talk about today. After his PhD, he started a pilot study financed by the Max-Planck Institute on these species, and then did a postdoc with John Wingfield, the father of the challenge hypothesis. And Wolfgang also did a lot to improve this hypothesis and to show other reasons that lead to increased testosterone levels in male birds. And since then, he has been working always on these topics with different species of birds, both in Central Europe, in Middle Europe, but also for more than 2 decades, he's been going back to Tanzania once a year to study life history in these Coucals. He published more than 140 papers in all journals of behavioral ecology, ecophysiology, comparative endocrinology, including also many papers in the proceedings, PNNS, eLife, and so on. And so it's really a great pleasure to have him here today presenting us his study on the competing females and carrying males, on the ecology and physiology of a reversal in sex roles in these bird species. So Wolfgang, if you can share your screen.
Sorry, I forgot to unmute myself. And when I share the screen, I can't see the Zoom screen anymore. So anyway, so I try to share the screen again. And I hope you can all hear me.
Yes, it works good.
Thank you very much. Yeah, then first of all, good evening, good morning, everybody. Thank you for being here to listen, and thank you, Carsten, Eduardo, and Lauren, and the FINE team for inviting me to give a presentation in this exciting series. Many famous people have been giving a presentation here, and I'm glad I can be amongst them today with and talk about Coucals, or competing females and carrying males, on the behavior and physiology of sex roles in Coucals. Now, okay, now the slide works. Sorry, always these technical difficulties in the beginning. So as Carsten already said, I have been interested in sex roles already during my PhD thesis, and I started with hyenas. And while doing my PhD, and I would recommend that every PhD student as well, I thought about what am I going to do next. So I was interested in sex roles, but I wanted to do my own study. And one of the things that I definitely wanted to do is go back to Africa. So I wanted to work on an African species. And then I stumbled across this paper, a small paper by Carl Vernon in Ostrich, about notes on the biology of the black coucal. So basically nothing was known about this bird, but Carl Vernon had observed 2 females, and one of these females seemed to be polyandrous together with 3 males. I thought this is really interesting, and this is how I got to the black coucal. And I'm really grateful that right after my PhD, Wolfgang Wickler agreed on funding a pilot study, because since nothing was known on this bird and since I had no experience on birds, I first needed to demonstrate that I can actually work with these birds, so catch them, tag them, and do things like that, before applying for a grant. And it was only because Wolfgang Wickler said, just go for a couple of months, try it out, and then come back, I could do that. So I think this kind of mentorship is really important. And I'm glad that I could experience that. So talking about black coucals, I would like to start with a very recent paper by Schuker and Quarnemo, who sort of provided a very nice definition of sexual selection. They say sexual selection is any selection that arises from fitness differences associated with non-random success in the competition for access to gametes for fertilization. The nice thing about this definition of sexual selection, I mean, it's not very new, but it doesn't talk about sex at all. And I really like this sex-neutral definition of sexual selection. And it sort of emphasizes the point that we should be open about whether sexual selection acts on males or females or both. However, if we look into the majority of animals, we see, well, most animals are reproducing sexually and they have anisogamy. So they have gametes that differ very much in size. So we have large eggs and small small sperm. And this means that there is a female biased pre-zygotic investment. And this usually leads to sexual selection that is stronger in males than in females. And this stronger sexual selection in males than females also has consequences for parental care. In animals that provide parental care, we typically have a female biased post-zygotic investment. And there's also feedback going back to sexual selection here. And this, again, also leads to sexual dimorphism, which usually is more elaborate in males than in females. And this has been shown in a recent comparative study by Janneke et al. In Science Advances, where they demonstrate that this is the general pattern that we find in animals. When we talk about sex role reversal, we think that some of these things have changed, and this is a very tricky term, and I recommend reading King and Arnesieu's paper in Evolutionary Biology, where they debate whether this is an appropriate term or not. It's difficult, but I think it's helpful as much as the term stress is in stress physiology. You always need to define it. From a human perspective, I think 100 years ago, this might have been an appropriate illustration of sex role reversal in humans. Nowadays, this is much more complicated. If we talk about sex roles in vertebrates, then I would first like to, I really would like to see you, but I can't see you anyway. So if we talk about sex roles in vertebrates, then there is a very nice comparative study by Reynolds, where we basically see that in most vertebrates, like reptiles, amphibians, and teleost fish, there is basically no parental care, with some exceptions. And you see that parental care in fish is male biased, and in amphibians it's approximately, so there's female only care and male only care, and there is biparental care, which is rare, but male and female only care is approximately similar, whereas in fish you can see that there is the more majority of fish, if they provide care, it's the males. So if you talk about sex role reversal in parenting and talk to fish, we would need a different concept than if we talk to sex roles in humans or in most mammals, where 91% of all mammals have female-only care, only 9% have biparental care. In reptiles, there is no male-only parental care known as far as we know. Let me go to birds. Birds are special because 90% of birds have biparental care, and only 8% of birds have female-only care. So this is really something special about birds, but also there it should be noticed that the majority of care in most of these birds is still done by females. So in many birds, males do not incubate. Only females do the incubation, and males then only provide food to the young later on. I would like to draw your attention to this 1% here. So in 1% of all bird species, there is male-only care. And in contrast to many other cases, like in many cases where in teleost fish where males care, this is often a complete reversal in sex roles. So this is not only parenting, this is also the competition within the sex, so that females are actually also more competitive. So examples, now the screen works. Examples include mainly shorebirds, where we have this complete reversal in sex roles. So here females compete for matings and only males provide parental care. And this is what I would call my definition of sex role reversal, where we have stronger female competition and male-only care. And this is mainly in waders, in the so-called charadriformes, all of which have a precocial development. So this means after the eggs hatch, the young leave the nest and are only guided by their parents. So they search their food for themselves. So I'm studying this sort of complete reversal in sex roles in cuckoos. And most of you, if you think about cuckoos, you think of brood parasites. And this here is the phylogeny of cuckoos. We recently heard a talk here in this series by Christy Reel on the anis, and they belong to the Cotophaginae. So these are cooperative breeders. And in fact, the cuckoo group is one of the most diverse group in terms of mating and parenting strategies. And we have actually 3 groups of cuckoos, the Neomorphine, the Cuculini, and the Phenocophaini, which evolved brood parasitism, but all the other cuckoos are basically nesting species that also provide parental care. And I'm here interested in the Centropodine, or the Coucals, which mainly occur in Africa and in Asia. So the main distribution is in Asia, about 30 species in total. And those are nesting birds, and they are considered socially monogamous and biparental. But overall, we know very little about these birds because they haven't been studied in detail. So I study African Coucals. And there are roughly 8 species, depending on which phylogeny you use in Africa. And I focus on 2 of them, the black coucal and the white-browed coucal. As I said, we know there's relatively limited knowledge about coucals, but the nice thing about these 2 species is that the white-browed coucal seems to be the most monomorphic and biparental coucal, whereas these previous reports by Carl Vernon showed that the black coucal is the one with the most extreme sexual size dimorphism and social polyandry. I think the comparison between these 2 species is a cool thing because it occurs in the same habitat, so they live in a similar habitat, they feed on similar food, they have a similar ecology, and this questions whether, I mean, they should also have a similar mating system. So I think this is a cool comparison because I can study 2 related species in a similar habitat at the same time. But many funding bodies and journal editors don't think so because it's a 2-species comparison and you shouldn't do 2-species comparisons. For all of you who may have similar problems, I recommend reading a recent article that I wrote together with Hubert Schwabel on this topic. It's termed "The Tyranny of Phylogeny, a plea for a less dogmatic stance on 2-species comparisons," because I think we can learn a lot from these 2-species comparisons. And in this paper, we sort of put together arguments why this may be the case. And later in this talk, I will present you an example why I think it is really necessary to do such comparisons on top of phylogenetic studies. I'm not questioning phylogenetic studies, they are really important, but I think we should also, we also need these more detailed comparisons among 2 or few species. Okay, I would like to start my talk with telling you something about the breeding biology of the 2 coucal species, and then later go into the role of hormones in shaping mating strategies. So this is my study site. It's in Tanzania, in the southwestern part of Tanzania, in an area called here the Usangu Plain. And here you can see a view of this area. It's a floodplain, seasonally flooded floodplain. So when the rains comes, a lot of water come into the floodplain, and there's very lush area with a lot of vegetation. And during the dry season, it's basically a desert-like area. How do we study coucals? So this is our main method to catch them, or basically our only method to catch them. We use mist nets. We stay very close to the nets because coucals are famous for leaving mist nets within seconds. So you have to be very close. And once the coucal is in, you need to run. As you can see, Ignas Safari, my former PhD student, who is now a lecturer in Dodoma, running towards the net to try to grab this bird. Once we caught them, we take measurements, we take blood samples and so forth. And before we release them, we put little radio transmitters on the birds because sometimes they are really hard to observe. They spend a lot of time in these grasses crawling around. And with the radio transmitters, we can follow them and find their nests and see what they do. So yeah, this is, you see our telemetry equipment. This is how we follow them in the field. And we also observe them. We either use a car or a ladder or something like that to observe them, because otherwise it's really hard to see them. So these are our basic methods. And with that, I would like to go into some basic measures that we take. And the most important one that I would like to point out at the beginning is the sexual dimorphism. So here you see the mean and the 95% credible interval of a body mass of a female black coucal and a male black coucal here. And you can see the females are much larger than the males. So if you look at the sexual dimorphism index, you can basically say that a male is roughly 60% of the size of a female, so 59% of the size of a female. And if you look at that in the white-browed coucal here, you see also here the females are slightly larger, but the sexual dimorphism index is much higher, saying that a typical male is 90% the size of a female. So this extreme dimorphism that we see in the black coucal, in the bird world, this is only sort of paralleled by jacanas, which are also, I mean, they belong to this Charadiformes group, and they are also sex role reversed. So the male provide parental care, females compete for matings. And this is the only other group of birds that have such an extreme reversed sexual dimorphism. If you see a black coucal, I hope now this works. If you see a black coucal, I hope you can hear it. So you can see here female black coucals here. And this is a typical situation, and if you wouldn't know anything about this bird, you would assume that this is a male singing, but in fact, males rarely sing. It's mainly the females that sing to attract males and to deter rivaling females. And it can really be serious. So while a female is singing and advertising her territory, she can be attacked by other females. And this here is a female that we caught after such a territorial encounter. And you can see that they can really fiercely attack each other and cause severe wounds to other females. So sometimes it can really get rough. If we compare that to the white-browed coucal, so this is here a pair of white-browed coucals, and this is one of the sounds that many of you who have been to East Africa probably know. This is a duetting pair of white-browed coucals. So here both partners sing, and they jointly defend territory by singing, which I have shown together with a collaboration with Henrik, where we investigated the song and what the duetting is used for in the white-browed coucal. On this side, you can see how many mates in a year a female black coucal or white-browed coucal have. And you can see here that, so here's the number of mates per year. And here for the black coucal female, you can see that the average black coucal female is mated with 3 males. They can have up to 5 males. And it rarely happens that a female black coucal is only mated to 1 male. These are minimum numbers because sometimes we really have difficulties in tracking all the mates of a female. So these are the minimum numbers of mates each of these females had. So obviously these females are polyandrous. You can see that most female white-browed cuckoos were monogamous, but there are a few that show polyandrous matings. And this was an interesting observation for us. We took a long while until we realized that there is some polyandry in the white-browed cuckoo female as well. And just to note, all of these polyandrous matings here are simultaneous polyandry. So these females are mated with several males at the same time. If we now turn to the males, on the left side, we have the black cuckoo males, on the right side were white-browed cuckoo males. You see that most of them are monogamous in both species. They are monogamously mated to one female in one season, but you also see some kind of polygamy in males. But all of this is sequential polygamy. So at one time, a male is mated only with one female, and if the female disappears or has been ejected by a rival, then the male may switch the partner, but normally it is monogamous. So this is, in case of the females, we have simultaneous polyandry. So at the same time, in case of the males, all of these polygynous cases, all of these polygynous cases are sequential. So here you can already see a huge difference between the two species. If you look at the clutch size, we find that the clutch size of these two cuckoo species is relatively similar. So the median clutch size is 4 eggs for both species, with a range from 1 to 7 in the black cuckoo and 1 to 6 in the white-browed cuckoo. So very similar. Okay, so here's something. Oh, it's just, I can't see it. So this is the egg volume. You can see that despite the fact that the female black cuckoo is on average larger than a female white-browed cuckoo, you can see that the average egg volume, the egg size, is smaller in the black cuckoo. And if you compare it for size, so here is the body mass of the mother compared with the egg volume, you can see that if you match white-browed cuckoo and female black cuckoos for size, you can see that same-sized female black cuckoos lay smaller eggs than same-sized white-browed cuckoo females. So we see this as an adaptation for laying more eggs so they lay more eggs, but smaller eggs, and the white-browed cuckoos lay fewer eggs, but larger eggs. If we now go to the behavior of the incubation, we see that in both species, the majority of incubation is done by males. So in the white-browed cuckoos, it's 80% of incubation. Whenever we come to a nest and look who is incubating, the male or the female, 80% of the time it's the male, 20% of the time it's the female. If you go to the black cuckoos, it's 100% male. Only males incubate the eggs in the black cuckoo. If we then look further, once the eggs hatch, what happens then? We look at feeding rates, and also here an interesting observation. So if you look at the white-browed cuckoos, then you see that females and males feed the young at a similar rate, whereas the black cuckoo females basically do not feed their young. There are some few exceptions, but normally they never feed the young, whereas males do all the feeding. And you can see that the average feeding visit per hour for a black cuckoo male is much higher and then the combined feeding rates of white-browed cuckoos, which is interesting. So they work far more, they work more than 2 parents for the white-browed cuckoo. So you can see again, generally socially monogamous species with similar sex roles, and here the classical or socially polyandrous species where the males are responsible for the care all by themselves. We suspected, because I mean we saw a lot in the black cuckoo, it's very frequently you see females copulating with males and not necessarily the male that she's actually providing a nest for. So we actually thought that there might be a lot of sperm competition going on. And something that we measured was the cloaca. Here is a cloaca of a male black coucal. And we found this huge difference between the cloaca size of the male black coucal compared to the female. So the male has a much larger cloaca than the female. And if you look at the same in the white-browed coucals, we find a much smaller difference. And also the white-browed coucal males have a much smaller cloaca than the black coucal males. So this was some indication that if there is something like sperm storage going on in the cloaca of the black coucal, that this would be an indication for sperm competition. And so the question is, are they actually feeding their own offspring? And then my PhD student, Ignas Safari, came in and he looked at extra-pair paternity of black coucals. And for nests where we had genotyped complete clutches, we found that 50% of nests contained extra-pair young, and actually 17.3% of all nestlings were extra-pair young. When we look at that in the white-browed coucal, we find that only 5% of nests have extra-pair young, and only 2.4% of these of the nestlings in these nests are extra-pair young. And the interesting thing is that all of these nests, so these 5% of nests, were nests from polyandrous females. So all females that were socially monogamous had not a single extra-pair young in their nest, but the socially polyandrous females in the white-browed coucals were the ones with the extra-pair young, which were, of course, from the respective other mate they were together with. And if you look at the number of extra-pair fathers, here is the number of extra-pair young per clutch in relation to the number of extra-pair fathers for the black coucal. We can see that most nests that have extra-pair young have one extra-pair father, but there are nests like they have two extra-pair that have two extra-pair fathers, and the most extreme case, I mean, that was just one nest here, where there were 3 extra-pair fathers for one clutch. So obviously, most of the time, it's just one male that is the extra-pair sire here for if there are 1 or 2 extra-pair young in the nest. But sometimes there are 2 or more extra-pair fathers per nest. If we look at something like individual reproductive rates, so here I show you the number of eggs per season, but it would be similar number for the number of young or hatchlings or fledglings per season. You can see that the average female black coucal has way more eggs than the average black coucal male incubates, or that the average white-browed pair lays or incubates. And this number here, so for the black coucal males and for the white-browed pairs, we can be pretty sure about these numbers. So these are good estimates because we find all of these nests. For the black coucal females, these are very conservative estimates because we are not always sure that we find all partners. So actually these numbers can even be higher. So to summarize this part of my presentation, so for the white-browed coucal, we found that females are slightly larger than males. They are socially and genetically monogamous. They do joint territory defense by duetting. Females and males incubate and provision the young more or less equally. The reproductive output is similar for females and males. And the variance in reproductive success is similar between females and males. But there is some flexibility in sex roles. So occasionally we find polyandrous females that mate with more than one male. And I think these are the most interesting cases. So overall, we could say that researchers say there is weak potential for sexual selection on females in this species. And as I said, it's the coucal species with the greatest similarity in sex roles as far as we know. But I mean, for many coucal species, so I think for 28 species of coucals, for the remaining 28 species of Coucals, we know very little, but so far this is what we could summarize from that. In contrast, the black Coucal, here the females are larger than males, significant and really larger than males, only paralleled by jacanas. Females are socially polyandrous, on average mated with 3 or more males. Females sing and defend their territory, sometimes very vigorously against other females. They are highly aggressive, and only the males incubate the eggs and provision the young. There is high extra-pair paternity. Actually, I think our extra-pair paternity rates are the highest reported for any socially polyandrous species. And the reproductive output is larger for females than males. And also the variance in reproductive success seems to be larger for females and males. I haven't shown these data, but this is also what we find. So the new Coucal Times could state that researchers say there is strong potential for sexual selection on females, but because we have this sperm competition among males and the high polyandry, also some sexual selection on males. And so far, this is the only known altricial bird, so bird that has young in a nest and that need to be fed in a nest, the only altricial bird with obligate sex role reversal. There are occasional reports for some woodpeckers, but not as fixed as this system seems to be in Coucals. So coming back to this initial quote from Schucker and Quarnemo and to the anisogamy and sexual selection paradigm, sometimes anisogamy and female-biased pre-zygotic investment leads to sexual selection, but this time there's stronger sexual selection on females. And there is male-biased post-zygotic investment, and there is more elaborate trait expression in females. And I find it highly interesting to study which factors are important in such kind of a reversal in sex roles. And important parts are, I cannot go into detail today because I would like to focus on hormones later on, but important factors are life history factors, and something that plays a role that has been shown by Malte Andersen's work is nocturnal incubation by males seems to be an important prerequisite for such a reversal in the parental care patterns. The ecology, it seems to be important that there are superabundant food resources so that one parent is sufficient for parenting in combination with high nest predation. And the sex ratio seems to be important. If additional males are available, then in white-browed Coucals, then white-browed Coucals become polyandrous. And in a recent comparative study, Luc Eberhard-Phillips has shown for plovers that if you look at the adult sex ratio here, you see that in the middle here, this would be an equal sex ratio. On the left side, this is a female biased sex ratio. On the right-hand side, this is a male biased ratio. And if you look to the top here, this is the proportion with parental cooperation, so shared parental care. And you see basically this is an inversed U-shaped curve. And you can see that the more female biased the sex ratio, the more likely the females provide parental care all by themselves. And the more male biased the sex ratio, the more likely the male provide parental care by themselves. This is a really cool comparative study published in Nature Communications, showing that the adult sex ratio seems to be connected to the parental care patterns in plovers. Now, the interesting thing for Coucals is that if you look at the hatching ratio of sex ratio of black Coucals and white-browed Coucals, we do not find a difference between females and males. So it's basically equal, the sex ratio. But later in life, we also see that the sex ratio remains equal in white-browed Coucals. But if we look at black Coucals, our study population has very high male biased sex ratio. So 75% of all adult individuals in our study area are males. So we have much more males than females. So we think this bias in the adult sex ratio plays a role. It is connected to this reversal in sex roles, but we do not know whether sexual selection causes an adult sex ratio bias, or whether the adult sex ratio causes a bias in sexual selection. It's a very tricky thing to find out, but it's very interesting. And since we have this equal sex ratio at the nesting stage, we would really like to find out what causes this change from being a nestling to being adult. We have studied juveniles so far. We don't really find much there. What we think might be important is migration. The black coucal is in contrast to the white-browed coucal, a migratory species. And here you see our study site. This is the rainy season. This is the dry season. Here in the rainy season, there are many black coucals. In the dry season, there is no or only very few black coucals because there is no habitat for them. The white-browed coucals remain mainly in these sort of bushy areas that have some water, but the black coucals, most of them leave. And if you look at the wing load of black coucals and white-browed coucals, we see that the wing load of females and males in the white-browed coucal don't really differ. But we see that because the female black coucals are so much larger than the males, they have a much higher wing load than the males. And they need to migrate. So what comes next is we want to study migration of black coucals. And the only thing to do so is with the help of Martin Wikelski, who is really a visionary and who managed to bring this antenna to the International Space Station, from where you can now track birds equipped with a small transmitter like this. So last year, we started to put these Icarus tags on black coucals, and we now have the first results. And a year ago, if somebody would have told me what is happening, I wouldn't have believed a single word of what this person would have told me. Because if any of you has ever seen a coucal flying, you wouldn't believe it would be able to fly more than 3 kilometers. This is what they do. Here is our study area in southwestern Tanzania. This is day 1 of the migration of one coucal. This is about 500 kilometers. This is day 2. This is day 3. This is day 4. And this is day 5 and 6. So from the southwest of Tanzania, it migrated to the northeast of the Democratic Republic of Congo, close to Lake Albert. And this is where this bird is still up to today. And I'm waiting for the next migration to see where this bird is going. But with that, I switch gears and come to hormones. Carsten asked me to tell something about hormones. The role of hormones in shaping mating strategies. So if we have something like, let's accept it for the moment, sex role reversal, often people think testosterone plays a role, and here you would assume that the female has higher levels of testosterone than the male, because the roles are reversed. I studied that already during my PhD thesis. I looked at testosterone concentrations in spotted hyenas. And there is a myth in the literature and in the minds of people promoted, I think, starting with Stephen Jay Gould and others, that female hyenas have higher testosterone levels than males. This is not the case. This is the female typical level of testosterone of a female hyena. This is an adult, reproductively active male hyena, and the levels of the males are way higher than those of females. But spotted hyenas may be not that interesting. They don't really have reversed sex roles. They have female-dominated societies, but they do not really have reversed sex roles. What about animals with complete reversal of sex roles with higher mating competition in females than males? Many studies have looked at that in socially polyandrous birds, and all of these studies have basically demonstrated relatively normal testosterone patterns. Up to 2004, nobody had looked at that in a sex role reversed Coucal. So we did that with the black Coucal. And here you can also see that here in black are the males and in white are the females. In the pre-mating phase, males have higher levels. During the mating phase, males have much higher levels than females. And when feeding young, males have only high testosterone levels because females usually do not feed young. So again, there's no reversal in testosterone in this sex role reversed bird as well. But together with Conny Vogt, we looked also into the brain of the black Coucal, and we found in a nucleus that is relevant for social behavior, the nucleus tenii, that there are more androgen receptors in female nucleus tenii than in male nucleus tenii. So not only the number of receptors, but also the density was higher in females than in males. So this basically means that in this particular area of the brain, females can react much better to lower concentrations of testosterone. So there might be some sex role reversal in this respect going on in the brain of black Coucals. We later on showed that progesterone actually modulates androgen-induced territorial aggression, but I don't want to go into this here now in detail. You can look that up in this publication if you're interested, but I would like to stick to testosterone. We also looked in female barred button quail, another sex role reversed species, into testosterone, and we compared the testosterone level to the size of this black throat patch of the females, and we found that actually the blackness and also the size of this throat patch correlates with testosterone concentrations, but you can also see here that the testosterone concentrations are really low. So there seems to be something going on with testosterone, and I then did a comparative study with John Wingfield to look at the testosterone concentrations of comparatively of birds with different mating systems. And what we basically find that there is basically no difference between polyandrous birds and socially monogamous or polygynous birds in terms of female testosterone or any kind of relationship there. So that there doesn't seem to be something special. But there is a problem with these kind of studies. And I would like to illustrate that with this example here. So here we have, for instance, a comparative study on egg size variation among tropical and temperate songbirds. This was published by Thomas Martin in PNAS. And to do such a comparative study, this is on the measurement basis relatively easy. So you have a caliper and you measure the egg size. And if you are a different study group, you may have a different caliper. So this one is made of plastic, this one is made of steel. But there may be some differences between these calipers, but if they are properly made, the difference will be small. So this will result in a small error. And you can easily compare measurements that have been collected by different groups. Now, if we turn to hormones, the situation is a little bit more tricky. So if these hands here simulate the antibody that you use to measure a hormone, then these two different antibodies here may be able to measure the target hormone. So both of these antibodies bind the target hormone. But then there's always some cross-reactivity, and you may think there may be a related hormone, and this antibody here may also bind the related hormone, whereas this antibody here does not bind the related hormone. So you actually find there could be a huge error. On top of that, these antibodies do not only bind to hormones, but there are also matrix effects. And the matrix effects may differ between the antibodies or the extraction techniques that you use, so that this antibody here may not have a strong matrix effect because of its properties, whereas this one may have. Again, this adds up and makes erroneous measurements. So if you now compare these measurements from different labs, this error can be fatal. And there is a very nice study by Fanson and colleagues in Methods in Ecology and Evolution that I can really recommend. What they did is they sent samples, plasma samples of birds with known amounts of corticosterone to 19 different labs around the globe, and let these labs measure these concentrations without knowing the concentrations, and then send the data back, and this is the result. So you can here see this is the actual concentration in nanogram per milliliter. And these are here the measured concentrations between the different labs. So this here would be the correct estimate, and you can see that some labs here are very close to this correct measurements, but some of them are really off. And this is actually an about 10-fold error that came out of this study. So any comparative study that wants to compare hormone measurements between labs needs to deal with this potential of a 10-fold error. So I give you some examples for comparative studies, and not to blame other people. I use comparative studies that I did myself. So this one was done with some colleagues in John Wingfield's lab. Here we looked at testosterone in tropical birds, the effects of environmental and social factors. And the main effect we found is the shorter the breeding season, the higher testosterone levels. So here you see the length of the breeding season, and you see testosterone concentrations. And corrected for phylogeny, you can see that there is basically a relationship here. You can also see that the difference is pretty high. So maybe this is a robust comparison, even if you consider these lab differences, which we also try to statistically control for. But so these are large differences. The other study are the data that I've just shown you. So this is male to female testosterone ratios. These are the data that I just showed you. Here we talk about very subtle potential differences. And I think to show subtle potential differences, these comparative studies, given the large error that we might have between labs, are not really helpful in resolving. This is why I think within lab comparisons of two or few species might be helpful. We have been doing that for the black coucal and the white-browed coucal. We recently added the coppery-tailed coucal, but we haven't enough data to compare that yet. So I want to show you our recent data on testosterone in black coucals in comparison to white-browed coucals. So if you look at males, we find little differences. In black is the black coucal, in white is the white-browed coucal. These are different breeding stages, and you can basically see the means and the 95% credible intervals. There is large overlap, so these levels of testosterone are very similar between those two species. This was kind of surprising because we know this is a cut open black coucal, and what you see here is its testis, and it's actually only one testis because black coucals only have one testis. So we thought, okay, if a species has only one testis, its testosterone levels may be low. Actually, that's not the case. The white-browed coucal has two testes, but obviously the testis size doesn't determine the testosterone levels. So even having only one testis, black coucal males can produce as much testosterone as white-browed coucals. But now what about the females? So here we find big differences. So here again in black, the black coucal females, and in white, the white-browed coucal females. And you see on average in every breeding phase for which we have comparative data, we find that black coucal females have twice as high testosterone as white-browed coucal females. So the black coucal females have higher testosterone. So what we saw in the brain, this higher sensitivity to testosterone, is also reflected in the blood. But this difference is large if you do that in a within lab comparison using the same methods of extraction and measurement. But if you do that, if you try to do that between different labs, you may not be able to see this difference, especially at these low levels of testosterone. So this is why I think it's really important to also have these more in-depth studies of two or few species comparisons within labs, especially for physiological measurements such as hormone levels. And with that, I would like to conclude that testosterone likely plays a role in shaping mating strategies of Coucal, but it's a subtle one, and it's only one that you can only detect if you closely compare two or few species. I hope we can add more species in the future. And these subtle differences are really difficult to compare across labs. And whenever you do comparative, phylogenetic comparative studies on labile traits such as hormones that are measured in different labs, you need to be very cautious and careful because you probably can only detect large differences. And this is why I really think we need a less dogmatic stance on two species comparisons. And with that, I think I am a bit over time. I would like to finish thanking a couple of people. So Liz and Neil Baker, Susanna Joos have been really important for logistic support in Tanzania. Funding came from the National Geographic, from the Ecological Society and the Max-Planck Society mainly. Also my PhD student here, Igna Safari, was funded by the German Academic Exchange Service. And I would like to thank him, who was an extremely gifted PhD student. Now he's a lecturer in Dodoma, and I hope he's developing a great career there. This is Puyo Makombo, my long-term field assistant, who I really owe much because I'm only in the field nowadays for 4 to 6 weeks, and he spends a lot of time chasing and observing Coucals. Without him, this would not be possible. And I would also like to thank Ingrid Schwabel, who has been sort of accompanying this project and other projects since they exist, and she has been an invaluable help in the field and also in the lab. I also would like to thank Puyo Makombo, the brother of Musa Makomba, the brother of Puyo, who has been my field assistant for many years. Luke Eberhard Hertel, with whom we recently developed models on the adult sex ratios. Felester Urasa, a local from the University of Dar es Salaam, my local collaboration partner, and Martin Lukelski for providing Icarus tags. And with that, I would like to thank you for your attention. And I think I do not need to make further further advertisement for ethology because Carsten has already done that. And I stop sharing my screen now, and I hopefully can see you now again. Yes, finally.
Yes, thank you very much, Wolfgang, for great, interesting talk with many different aspects from life history, behavioral ecology to ecophysiology. Like always, when you have a question, then please type a question mark in the chat function, and then I call you up one after the other. And when I call you up, please first quickly introduce yourself. What's your name, where you're coming from, what you study, and then ask your question. And I think already during the talk, there was a question mark by Maren Hook.
Yeah, thanks for the great and very exciting talk. Actually, you answered my question because I had the question when you were talking, when you were saying that on average, females have more eggs. I was thinking of one of the papers by Hannah Cuckoo. It takes two to tango.
Always takes two, yes.
With a reverent bow to her. Unfortunately, I can't be there next week. But so the question was essentially answered because you said they have a very imbalanced sex ratio and gave some potential causes for that. So I have a question out of ignorance. What's the matrix effect? I didn't quite get it when you explained it.
The matrix effect is basically, you know, I mean, you have a solution with an antibody, and then you pour in bird plasma or any kind of plasma that you have extracted, but there are other substances like lipids, proteins that might interfere with your binding. And that can differ between antibodies, and it can also differ between extraction methods. This is the matrix, the background, sort of. It's the background. I just used this cinema thing to illustrate it.
Thank you very much. And I forgot, so I'm Maren Hook and I'm from the University of Derby.
Nice to meet.
The next question comes by Eduardo, please.
Thank you. Thank you, Wolfgang. Very, very interesting talk. Lots of questions. The resemblance with some of the primate species that we study are quite interesting. And so, and it's related to that. I was hoping you can comment a little bit on the natural history of the Coucals as they go from leaving their nests to becoming one of those reproducing adults. Because what maybe I didn't follow you well, what we have and really remarkable similarities with what we see with the pair living owl monkeys. Why couldn't we have still strong selection pressure, but the pressures be similar in males and females? We see a lot of competition in owl monkeys, but we do have a floater stage. So I was curious, how do the young adults get to be reproducing adults in the Coucals' life history?
Yeah, that's a tricky question because we don't really know. I mean, I had this discussion before with Carsten that we have this. This is also why I really want to know what's going on using these Icarus transmitters. We have a return rate of black Coucals, which is less than 1%. So they leave the study area and they basically never come back. And I don't know what is happening with them. Are they all dying? Are they breeding elsewhere? And I think the Icarus tags are the only way to find out what is going on. What happens with the juveniles? So we have tracked juveniles. So they leave the nest after two-week nestling phase. Then we call them groundlings. So they cannot yet fly. So they leave the nest because this nest is a dangerous place. You can get predated by snakes or monitor lizards or whatever. So they leave the nest at a relatively early stage before they can fly. And we find that in this groundling phase, which is approximately another 2 weeks before they start flying, we find higher mortality in female black Coucals than male black Coucals, whereas in the white-browed Coucals, the mortality between males and females is similar. But the difference in this mortality is subtle. So it doesn't account for this huge difference in adult sex ratios that we find later on. It may account for something, but not all of it. And so, I mean, there may be one explanation necessary for that. So, I mean, you saw that there is this big size dimorphism between female black Coucals and male black Coucals. And this size dimorphism already starts to develop in the nest. So the females leave the nest at a slightly higher body weight than males. But relative to what they have to reach as adults, they have a far more longer way to go. So they leave the nest at about 40% of adult body size, whereas males leave the nest at about 70% of adult body size. So relatively speaking, they leave the nest at an earlier developmental stage. And this may be the reason why there is more predation on females after leaving the nest. But it doesn't really explain the big difference that we find later on. And then after that, we don't really know much what's going on. So all we see is the following year, we see that there are new adult birds, but the major number of them, I mean, with few exceptions, I mean, we get completely excited if we see a ringed bird. Most of them are unringed.
Thank you.
Okay, so first of all, I would like to encourage also our younger colleagues, the students, PhD students and postdocs, if they have some interesting questions. I'm sure you have, don't be shy to ask them. But the next question comes from a slightly older colleague, Michael Taborski.
Yeah, Michael Taborski from the University of Bern. Thank you, Wolfgang. This was really interesting. You opened a lot of very interesting general questions. And the most obvious question to me is, of course, why males invest so much in care in the black Coucals. And as you advertised this comparison between two closely related species sharing the same ecology, I think you could perhaps provide us with a bit more information about your ideas, what really could cause this specialization of males in brood care in the black Coucal. Especially, you mentioned that nest predation could have an effect. So my question is, how do you think nest predation can cause a specialization of males in defense, especially if the females are the larger sex and perhaps also the more efficient repellers of predators? And especially in the comparison between these two species, how can nest predation explain the bias towards males in black coucals in brood care?
So yeah, so one of these, I mean, this is really, I mean, this very much goes into a just so story. This is how I imagine it. I don't really have proof of that yet, but I mean, the idea goes along these lines. And these are also ideas that are, I mean, they are not originally by me. I mean, Malte Andersen has worked a lot in that direction and formulated these hypotheses. So if you have a situation with high nest predation, especially during the egg stage, then if this nest predation is high, it is maybe advantageous for the female to sort of continue collecting resources to be able to relay a replacement clutch when the male takes over most of the incubation, because then she can soon, especially if you have such a short breeding window as a black coucal, then she can soon provide a replacement clutch. But in case the nest is not depredated, the female still has the resources and could share these resources with a different male if these males are available. So that's one of the ideas regarding nest predation. And we actually find a difference in nest predation during the egg laying or the incubation stage between white-browed coucals and black coucals, because they share a lot of things, but there are also a lot of differences. And one of the major differences is that black coucals build the nest in high grasses, and they are basically unprotected, especially from snakes and monitor lizards or some mongoose who frequently predate on them. Whereas the white-browed coucals, they almost exclusively build their nests into acacia bushes, where they are much safer from predation. This is the best what I can offer on that.
So I derive from this that there are much more replacement clutches in black coucals than in white-browed. Is that right?
Yes.
Okay. Yeah. At least I mean these are data that point in this direction, of course. Thank you.
The next question comes by Eckhard Heimann.
Yeah, I'm Eckhard Heimann from the German Primate Center, and I was particularly fascinated by your talk because I have been working together with Maren on one of the few mammals that have a polyandrous mating system, namely tamarins. And my question refers to you mentioning that females are defending territories. Are they actually defending a space, or are they eventually defending males?
No, they are really defending a space. It's really a territory. So you can also see these border encounters between females. Females never follow males or something like that, or protect their males from other females. It's really a territory that they defend.
Okay, thank you.
So the next question comes from lecture hall, Vario main lecture. I cannot read all of it, but you will probably know that it's you.
Yeah, sorry, actually it was me, but in another room. We had to leave the other room, and I posted my question a bit later, but it's okay, I can ask it again. First, thank you for this talk. It was really, really interesting. So yeah, I'm Marion Delinger from the Department of Aquaculture and Fish Biology in Iceland, and I'm studying eco-evolutionary patterns of development of spatial cognition and personality in the Arctic char. Yeah, and I wanted to do a special hi to Batis Sadughi because we were colleagues in vet school, and I was surprised to see him here. But yeah, and my question was, maybe you said it before, but I was a bit in a hurry leaving the room. could it be possible that the bias for the sex ratio for the females could be a result from the fact that the females are the ones singing and the one defending the territory, so they are making themselves being very conspicuous to the predators? Would and maybe then they get caught more easily by the predators by behaving so conspicuously? Would it be possible?
It's a possibility, and with Luc Eberhard Phillips, or Luc Eberhard Hertel as he calls himself now, we are just looking into this. So we have roughly 20 years of predation data during the breeding season, so we can compare predation on females and males. So I agree, females expose themselves, and we see them being attacked by potential predators. But the same happens to males while being on the nest. We have quite some deaths of males while incubating, while feeding their young, being depredated at the same time with their young. So I'm not sure who wins that battle, but we are currently looking into that.
Okay, thank you.
So then the next question is by Baptiste Sadushi, please.
Good evening, thank you. And I also say hi, Marion. Thank you very much, Mr. Goymann, for the talk. I'm Baptiste. I work in the University of Göttingen and mainly on behavioral endocrinology. And so I was very interested by especially also the second part of your talk, and maybe taking a bit of a step back on all the things you mentioned about testosterone. After all, what do you think is the role of testosterone in either female dominance, expression, or sex reversals? Because you mentioned the hyenas, males still have a higher testosterone. We know in lemurs that some papers are trying to report that females have a higher testosterone, and that might be why they're dominant, but we see that apparently it's not necessary. And now in the black cuckoo, Coucal that you presented, they still have lower than the males, but you talked about this higher receptor density in the brain, and then you went on to present the fact that they have higher testosterone levels than the other the females from the other species, kind of hinting on the fact that it could maybe still be linked to their male-like behavior in the black cuckoo. So how do you wrap all these things together, and what's your personal take on it after so many years of thinking on those questions? Thank you.
Yeah, it's a tricky one. I mean, with regard to hyenas, I mean, there are the hyena groups from Berkeley and also from Michigan. I think they have demonstrated that androgens definitely play a mechanistic role in some aspects of female masculinization and so forth, but mainly this is through androstenedione, especially late in pregnancy. These levels are very high. So for sure, there is a role of androgens, maybe not necessarily testosterone, but androgens in this specific pattern of female hyenas. For the black cuckoo, I mean, with our progesterone experiment, we have shown that we started this experiment basically with the idea of the challenge hypothesis. So we wanted to challenge females with a simulated territorial intrusion to see whether testosterone changes. And to our surprise, we found that testosterone does not change, but we found that progesterone went down. And then we thought, okay, this is cool. And there were similar observations from female mice. And we thought, okay, what happens if we implant them with progesterone? And so we keep progesterone levels high, and this actually led to a lower aggression. So progesterone was sort of modulating that. In mice, it has been demonstrated that progesterone is exactly doing that, modulating aggression between females. That is induced by testosterone and estradiol. And my hypothesis would be that something similar is going on in the black cuckoo, that testosterone and maybe estradiol, which is basically always at non-detectable levels, it's hardly detectable at all, but that these are the players modulated by progesterone, and that we have these special areas in the brain that are particularly sensitive. And by that, females just avoid having high levels of testosterone that may be otherwise detrimental to their reproductive system by just increasing the sensitivity in relevant areas in the brain. That's one idea, but it could also still be that, and that would be an interesting experiment to do with the white-browed cuccals, if we would increase female competition in the white-browed cuccals, maybe they would end up with similar high levels of testosterone as female black cuccals. So it could, maybe it's not the hormone driving the behavior, but it's the behavior driving the hormone. This is what we don't know, and this is what would need to be tested. It's just, I found this result very interesting, that we find these subtle differences. Of course, this is now just from 2 species, and it would be interesting to do the same thing for maybe a couple of shorebirds or include other cuckoo species. I mean, we have started with the coppery-tailed, but the numbers are really low, so we will take a couple of years until we get enough samples to look at that. Whether this is a consistent pattern, and maybe do experiments, especially in these species where these levels are lower, to see whether you can actually get them up with more competition. So I think it plays a role. I mean, it has to play a role. I don't think they have these receptors in the brain for fun. I mean, for sure, there's a reason to have these receptors, and I assume that testosterone binds to them and does something. It's just not my, I don't want to go further. I mean, I killed a number of females to look at their brains. I'm honestly done with killing females and birds in general, I hope, unless I have a very good reason. This is the level at which I can go. I don't want to go much further. I can do further behavioral experiments, but I won't kill more black cuccals or white-browed cuccals for my research.
Thank you very much.
And we have another question by Eckhard Heymann.
Yeah, sorry, me again. You mentioned female competition as one factor in sexual selection. And given the heavy investment of or involvement of males in paternal care, is there also potential for male choice of females?
Choice is always difficult to show, especially in a field study. I mean, what we observe in the beginning of a breeding season, when we start catching males, we catch these males in the area of one female or in the territory of one female, and then later on, when reproduction actually starts, we find this male in another female's territory. Whenever we catch males later in the season, they remain in the same area. So I imagine males coming to the breeding site, shopping around maybe for territories or females, and looking around whether there might be. I guess they probably look more for the territory quality rather than the female itself. But this is just an impression I have, and from these data that we get initially in the breeding season, when we catch males and we see them frequently later changing these territories, whereas later on they do not sort of switch sides. If there is male choice, I don't really know. I mean, it's really hard to get these data without sort of doing a choice experiment, which we haven't done in the field.
Then we have a question by Kaya Tombak. She wrote it in the chat, but I think she's here. Can you ask the question yourself, or you want me to read it? Kaya?
I can try. My internet's a bit shaky, but basically, with regard to the skewed sex ratio, is it possible that females just take longer to mature to reproductive age and to get big enough, and therefore might have a greater chance with that much more time, you know, out of a partnership to die before reproduction than males, and might that contribute to the sex ratio?
That's for sure a possibility, as long as we are not able to recapture birds or know where they go after the breeding season. We cannot really find out, but for sure this is a possibility that females take longer to mature.
Would there be a way to age them?
Yeah, actually we have done that, or we have sort of come up with a way of aging them without really being sure, and that actually points into such a direction. So there are plumage patterns. So like, you know, the juveniles, they have these barred primaries and secondaries, and also the coverts are barred. And we often see birds coming to the field that have partially barred primaries and secondaries and completely barred coverts. And we assume that these are first-year breeders. When birds come back with the primaries and the secondaries completely rufous, but the coverts still barred, we assume that these are second-year birds. And when the whole wing is rufous, we assume that they are at least 3 years old. There is some reason from the few molting data that are available from Coucals that this aging is reliable. I'm not 100% sure, but if this is the case, then we actually find a pattern that we have approximately equal numbers of first year, second year, and 3+ year males in the study area, whereas we have very few first year females in the study area. So the majority of females is 2 or 3 years old. So this would hint into such a direction that the first year females do not come to the breeding sites but stay elsewhere. That's a possibility, but we are not 100% sure about this aging.
Thanks so much, and thanks for a great talk.
Welcome.
Then we have a question by Qingyu.
Sorry, my camera is not working. I'm Jingyu. I'm a PhD student from the CNRS in France. So you mentioned the extra pair paternity in the black cuckoo. So I'm wondering how does the first male would treat the male that comes after? Does it be aggressive to those male like some polyandry species?
Yes, the males are sort of, I mean, you know, the typical situation is the following. So when a female starts to provide a male with a clutch, the male follows that female most of the time. So it really tries to stick to the female, but occasionally there are other males that try to drive away this male. So you see males chasing each other, and this is why I think there is competition between males. But as soon as 1 or at most 2 eggs are laid, the male starts incubating the clutch, and then he can no longer follow the female. And this is when the extra pair paternity happens. So the mate guarding has to end because he needs to incubate. And I think this again has to do with the predation probability. They start incubating after the first egg already, while the female still produces eggs for him. And if you look at the the sequence of eggs in relation to extra pair paternity, we can follow that because, I mean, the young then hatch asymmetrically. So we can, by the size of the young or by the hatching order, we can tell which are the first laid young or laid eggs and the last laid eggs. And we actually find that the extra pair paternity mainly happens at the later end. So the later laid eggs are the most likely ones that are extra pair. And this is actually the situation when the male is no longer able to mate guard the female.
So it's not like the male doesn't want to chase away other males. It doesn't have the ability to do that.
Yes, he needs to incubate, so the female can do whatever she wants.
Okay, thank you.
So there are two more questions in the chat, and I will first take the one from Maren Hook, as she says it directly relates to the one from Jingyu, and then after Maren will be Sulema.
Thank you, and sorry, Sulema. Yeah, I think I'm still a little bit unsure about how this is all happening. So does the female have several nests? And if she has only one nest and lays the eggs from several males in the same nest, what are doing the other males that are not providing care, or are they all providing care at the same nest?
No, each male receives his own nest. So the female first starts out with hanging out with one male. She builds a rough nest. The male then finishes the nest and she lays eggs. And she continues laying eggs into this nest until she thinks, whatever, this is a full clutch. And then she goes on to the next male and provides this male with a nest, and then to the next, and eventually back to the first, and so forth. So each male has his own nest and provides care for the young that are in this nest, whether the young are his or somebody else's.
Sorry. Then next question by Sulema.
Yes, hi, Sulema Tang Martinez, University of Missouri, St. Louis. And I'm having problems with my connection today, so I may interrupt in the middle. I guess my question also relates to the previous ones on extra pair paternity. I'm assuming that for the black Coucals, when you say that they have greater extra pair paternity, you're talking about males that do not necessarily have nests. In other words, let's say a female mates with 3 males and lays eggs for those 3 males. When you say extra pair paternity, it means that she's mating with a 4th or a 5th male that she does not lay eggs for. And I also wanted to confirm that I understood correctly in the last answer that males do in fact end up at times taking care of young that are not their own. I had another question about testosterone, but I think I'll just, hold on to that one. Thank you. A great talk.
Yeah, thank you very much. And yeah, this is also an opportunity to clarify that. So it's not the case that this is, defining extra pair paternity in polyandrous systems is really difficult. So I take the perspective of the male. So from an individual's male perspective, he is monogamously mated to a female, and any egg or young that ends up in his clutch that is not his is extra pair. And most of the time, this extra pair young or egg is from one of the other harem males of the females. Very rarely we have extra pair young from other males. This happens mainly from neighboring males, so males that belong to neighboring females. This also happens, but much more rarely. So in essence, if you take a female perspective, this is much more difficult, because from a female perspective, all the young of all males in her territory are within young. But for I think for this system, it's essential to take this male perspective, because the males do the work for young. They are not genetically fathers of. I hope that answers your question.
Yes, thank you very much.
Then another question by Eduardo, please.
Yes, I'm just hoping that you can help me with neuroendocrinology 101. So please, I excuse my naivete, but I continue to be very troubled. All of us have done some research measuring these hormones with colleagues, collaborators who know much better than I do. So you have shared with us some of the caveats of making inferences from measures of what we think is testosterone from some kind of medium where we share it in relationship to the receptors in the brain. And that I understand. Ourselves, because we have found levels of testosterone in female fecal samples that are higher than the levels we find in male fecal samples. One of the things that we're in the process of doing is sending some of those samples to do some mass spectrometry to really understand what is it that we're measuring, which I think is speaking a little bit indirectly to what you've been describing. Now, for those of us who cannot really go into the brains of primates, we can or we don't want to, how do we know what the receptors are for? Because how does one get to learn in these systems what kind of receptors they are in the brain for their various range of androgens that may be circulating in the bloodstream? So that's where I get lost. Let's say that I confirm that, yes, indeed, we are measuring testosterone, but I don't know what's being read in the brain.
So the thing with fecal samples is really complicated. So fecal endocrinology 101, you never can compare female fecal testosterone metabolites with male testosterone metabolites. You can't do that. It's impossible. I've shown that in a couple of publications, and I would really recommend reading. I have a Methods in Ecology and Evolution paper published in 2012 on the problem of sex and metabolic rate and many other things in fecal samples. So you just cannot do it because, you know, there's a lot of things going on, and all these antibodies are binding to many things and many metabolites. And if you imagine, I mean, blood is already a quite tricky and difficult substance. I mean, there are lots of things in there that can sort of affect your binding. But blood has a big advantage. You have the messenger there. What you see is the messenger. So the testosterone molecule in the blood is the messenger that does something in a target organ, be it the brain or something else. The feces is something that happens very late, and you do no longer have the target hormone. So what happens is the target hormone is at some point metabolized in the liver, excreted into the gut, and then heavily metabolized by your microbiome. And I would assume that depending on how your microbiome is sort of made up, the metabolites will differ. And it may be even, I mean, I've been asking that question, and none of the fecal persons has done the experiment. And I think it needs to be done because it's essential for this kind of field, that actually the composition of your microbiome might influence what you measure in the end. Because if you have a bacterium that sort of metabolizes testosterone into one hormone, and another individual has a bacterium that metabolizes testosterone into another metabolite, and then you come with your antibody that may be able to detect this one, but not that one, you may end up with a measurement that doesn't tell you anything about the original hormone concentration. And this is really something very tricky in this field, and I think it hasn't been, in my view, I mean, I stopped doing fecal analysis because I just don't trust them.
This is, of course, very, very interesting. I imagine from what you're saying, we have done fecal samples. What we have tried to do as well, which I imagine it's at least one tiny baby step in trying to address the challenges, is if you can validate across media. So if you're measuring in blood, in feces, we haven't done it. I mean, now I'm thinking cortisol now, where we have thought about really, we have samples, hair, blood, feces. So maybe, of course, it doesn't get us to measuring the messenger, but it may allow you to at least reduce to some extent the uncertainty that comes with feces. But I hear you. Thank you so much. Of course, it's extremely problematic.
You know, I mean, there have been great, like, I mean, there are all these validations that you can do, like do these hormone challenges with GnRH for testosterone and ACTH for corticosterone. So a lot of things can be done to make these measurements better. And I think these measurements probably work best in something like a cow and a goose that sort of has a constant input of food and a constant output of food. So you have sort of a relatively thorough digestion. And if you have one group of animals that sort of share similar food sources, you may assume that the microbiome is similar. So in these cases, it may work quite well, but I think it doesn't really work if you compare males and females, because they may have a different metabolism of the hormones. It may be okay to compare males amongst males and females with females, but between the sexes, I think it's basically impossible to do these comparisons. Within a sex, I think you can control for many things like the environmental temperature, like the season and things like that. So I think you can do a lot of things. And if you look out, I mean, there are many studies done with fecal analysis that at least make sense. So I think there is something there. But I still think that one of the experiments that if I would still be working in the field, I would do an experiment by changing the microbiome and see what effect that has on the excretion of hormones. I think this is an experiment that needs to be done.
Thank you so much.
Well, it's interesting, especially the microbiome might also change seasonally. So then you don't know, do you measure seasonal changes in microbiome or in hormones? Then comes a question by Irit Aloni, please.
Hi, first, thanks for a great lecture. It was very interesting. I'm a postdoc at Parental University in Israel, and I was wondering what does a female do while the males are nesting and taking care of the young? Do they still defend their territory? And also, would other females attack males or young of the nestling of in another territory? Thank you.
So, given the unlikely fact that all males of a female are busy with incubation or feeding young, then the female would still very much advertise. So she would sing a lot to attract further mates. Most of the times, the females are busy with the males they have because one may have lost his clutch, or the males are already, you know, we have situations that are really curious that males are still, I mean, their young have just left the nest. The males are still feeding these nestlings and they are already receiving a new clutch by the female. So they already start having eggs again and then I don't know how they do the incubation and the feeding of the young in combination, but somehow they manage to do that. So usually the females are always busy with one of their males. In the case of, well, sometimes we have females that have only, well, very rarely we have a female with only one male or with two males. And if all of these males are busy, then the females, they just sing the hell out of their throat.
Irid, we cannot hear you.
I'm saying wow, and would females also attack other territories or other nests? Would they only try to attract the males, or would they also try to destroy existing nests?
We have only anecdotal evidence of that. We have anecdotal evidence of females taking over other females' territories, and we find killed young at the nest without being depredated, and this is very unusual. So normally if a nest is predated, the young are eaten, but we found in these cases, we found nests with killed young. And we also find a situation that when males are feeding their nestlings and the female took over the territory, the female always tries to follow the male, and the male desperately tries to avoid going to the nest as long as the female is close. This is evidence we have, anecdotal evidence we have, that females might actually sort of use a lion strategy to increase their own reproductive success when they have taken over a territory of another female.
Thank you. Thank you very much.
And we have a question by Adriana.
Hi, I'm Adriana Maldonado from Universidad de Rosario in Colombia. Thanks, Wolfgang, for such a nice talk. I'm curious about the male dynamics. So I have kind of like two questions in a single one. So when they are choosing their territory or / female, because my understanding is that we don't know too much about mate choice, but let's say they pick the territory. Is there like aggression between males as for who takes the territory in the first place and then who settles? And then is there some, like, I don't know, some sort of rule that tells, okay, you can settle here with me, or you're not allowed to come and join this harem? And then once the, let's say, harem it's already in place, have you seen any kind of cooperation among males, like helping each other, taking care of the nest that is not their own?
It's really hard to say something about the dynamics with the kind of data that we have. In terms of competition, so I mean, if you imagine a female territory, then it's not that the female shares a territory with the whole territory with all males. So each male has a subsection of that territory that he normally does not leave. You see male encounters at the borders, mainly when one male is following the female and the female is crossing the area to the other male's border, then this other male will come and try to chase that male. This is what we typically see. But it's not like this is a communal territory, but it's for the female, it's a big territory, and for the males, they have sub-territories within that they normally work in, and no, they do not cooperate. So they only tend their own nests.
Okay, thank you.
And another question by Baptiste, please.
Yes, thank you. Sorry, I jumped back on what you were saying about the microbiome and hormones. Very simple question, but given that, for example, I may not have access to an experimental setup in the wild, in a wild population, do you think a parallel measurement of fecal and urine cortisol could be a first step in trying to analyze the impact of the gut microbiome, or no? And if no, why? Like, what do you identify as another problem in this?
I don't know. I mean, the problem is that species differ in the proportion of what they excrete into what. So I don't know if you can tackle that, but I mean, it might be a it might be a way to get a first hint, but I think the best thing to do would be to do something in the lab and give antibiotics or something like that to sort of destroy the microbiome and let it restore, or yeah, something like that. I think this would be much easier to tangle in a lab study than in the field.
Yeah, thank you very much.
Then probably the last question by Anthony.
Hi, this is Tony DeFiori from the University of Texas at Austin. Thanks, that was a really wonderful talk. And my question is related to the one that Adriana asked about cooperative or mutual behaviors that males might perform as a service to the females whose territory they're on. And I think you kind of answered that question at least a bit. And maybe I missed this, is there any evidence that the males in an area are related to one another? Are they coming from, because I think you have genotype data, right?
Yeah, we have parentage and relationship data between the males, and no, they are not related.
Thanks.
Okay, then I would like to thank Wolfgang again for a fascinating talk with so many facets, and all here in the audience for your lively contribution to the discussion. It was really a pleasure. And I would like to apologize to the YouTube audience that I will only be able to upload this video later tonight. I don't know what happened, but I will also tell my co-hosts if I find out what's the reason that I failed so today. With this, I stop the recording.